Hello, my name is Oleg Ermolinsky, and this is my student seminar for the Biocommunications Fall 2023 course. And my student seminar is about the induction of bacteriophage lambda's lytic cycle by RecA. All right, so bacteriophages are submicroscopic intracellular obligate parasites of bacteria. That is a bit of a mouthful, so we can break that down real quick. Bacteriophages are just viruses that infect bacteria. The question then arises, what are viruses? They are just genetic code, most oftentimes being either double-stranded or single-stranded DNA, though they can also be RNA viruses as well. And this genetic code is encapsulated in a protein capsid, which protects it from any sort of chemical or physical damage. There's also several types of accessory structures. In this case, in this image to the right, you can see a tail, there's also legs, and sometimes they even have spikes. And they're just accessory to the infection process, which I'll explain further on. And because of the simplicity of their structure and because of their size being submicroscopic, meaning you're not able to see them under a microscope, here you can see a size comparison where these viruses and phages are similar in size, are smaller than cells, smaller than bacteria, and even smaller than many of the organelles that our cells have. They're just simply incapable. They have no space in their structure, incapable of any sort of metabolic process. And because of that, they are obligate intracellular parasites. They require a host cell to carry out many of its metabolic processes that are needed for replication. They're incapable of their own replication, for that matter. And then that can go into a discussion of whether they're alive or not, but that is for another day. Interestingly enough, there are more phages on Earth than all organisms combined out there. And even more interesting, in the ocean, they compose the most biomass in of all the organic material present out there. So you take all the phages, there's more biomass of phages than everything else combined as well. To the right here, we have bacteriophage C4. It is one of the most well-known and well-described phages. It's a very simple phage, both structurally and genetically. It is from the Malviridae family. This to show that, these phages also have families, the same way we describe them in species. And they're also based on the same aspects where we look at morphology and genetics. Bacteriophages can undergo two different life cycles, either being lytic or lysogenic. And the lytic cycle involves once the phage has injected its DNA into the host, that DNA, the genes that are present in it, are expressed, and the DNA itself is replicated. And once enough DNA and the proteins are made, they form back into the virion. So you can see here, we have the DNA being inserted and the proteins being expressed to form, the capsid, the tail, the legs, and the DNA itself is replicated. They form back into the virion, and typically they also have a gene to express certain proteins to cause lysis. And lysis occurs, and the virions are released and are able to further then infect other cells. There's also the lysogenic cycle, where once the DNA is inserted into the host, that DNA then becomes a prophage, where it is retained either in the chromosome or also episomally in a far more rare case. A famous one being P1. It does this. It looks almost like a plasmid where it replicates in the background. But whether in the chromosome or episomally, as the host replicates its own DNA, the prophage is tagged along and replicated in as well. And that way, that the phage still is alive, you might say. And then temperate phages are the class of phages that are able to undergo both the lytic and the lysogenic cycle. So if they undergo the lytic cycle, they stay in the lytic cycle, but if they switch to the lysogenic cycle, for whatever reason, they can stay here and then go back into the lytic cycle. That's what temperate phages are. Now, most temperate phages are able to be induced by mutagens, mitomycin C and UV. What I mean by induced is that exact process that I had just mentioned just a second ago, where lysogenic strains, once they're lysogenic, are induced and become lytic. They switch over from the lysogenic to the lytic cycle. And what I mean by mutagen is any chemical or physical agent that can cause a mutation in some organism or whatever biological thing that has DNA in it. Mitomycin C is a drug that is used for chemotherapy. It blocks protein expression in cancer cells. And in prokaryotes, it is a potent DNA crosslinker. So a DNA crosslinker is any agent that forms covalent bonds between any nucleotides. So hopefully you remember that in DNA, the nucleotides only form hydrogen bonds between the two strands. So a DNA crosslinker will form covalent bonds between either adjacent nucleotides or between the two strands. They will form covalent bonds, and that is very harmful to the cell. UV is also known for its mutagenic processes, and it is best known for their thymine-thymine dimers, where two adjacent thymines form covalent bonds with each other. Now, it has been described even that in prokaryotes, a single DNA crosslink can prove lethal to the survival of the cell. And as such, that's why mitomycin C and UV are both considered like antibiotics against bacteria, that they're very harmful to the cells. And coincidentally, in the presence of either temperate phages, or more specifically, a strain containing a temperate phage, becomes lytic. So to the right, we have a figure here. This is actually from a lab that I have been part of for the last couple of years, comparing the growth of 3 strains. So you can see the growth or the concentration of cells by the OD600. And 1 strain is the wild type, and 2 strains contain phage. And under the induction by mitomycin C over time, the wild type strain, even though mitomycin C, as I said, can be harmful, they're able to survive or amend with the effects. And the 2 phase strains, as you can see here, there's lysis that occurs. So the cells are dying, they're being killed off by the phage that is within them because it is going back into the lytic cycle under the influence of mitomycin C. So phage lambda is an extensively studied model of temperate phages. So phage lambda is an E. coli coliphage in the family Cyphiviridae. And these terms are here to remind you that even though phage lambda is not an organism, it is not a species that we can classically describe, they can still be categorized based on morphology and based on genetic similarities, in this case being coliphage for its structure. This phage was first found in 1950 by this wonderful lady, Esther Lederberg. She was actually researching what we had just described previously with the UV effects on cells. She was studying the effects of UV being applied on E. coli, and on agar plates, she would notice that there would be clearings, spaces of lysis, and she would look into it further and find that she was actually inducing the phage, just as we had described previously. She was inducing a phage into the lytic cycle, causing that lysis, and then that phage would ultimately end up being termed lambda. The genome of lambda is 48,500 base pairs, which is far smaller compared to the host, E. coli, which has 4.6 million in comparison. And I say that to remind you that these entities, these biological entities, phages, are far simpler and far smaller than many of the species we study. And because the genome is far smaller, it can be easier in a way to study the genes present. There's less non-coding sequence and more genes are present with more function in them. You only have so much space in the capsid to fit, so every gene has to have a function. And because of this, it became a model organism in quotation marks because it's not alive by our classical standards. But because it's far simpler and it can be easier to track every gene and its function, It was actually used for many of the early discoveries in bacterial genetics and molecular biology. Many of these discoveries later being applied to actual bacterial genetics and not just bacteriophages. All right, so the maintenance of lysogeny and lambda's lysogens depends on C1. And I'll explain that real quick right now. So after the infection of a cell with lambda, the two genes CRO and C1 in italics meaning that it's a gene, determine which life cycle is chosen based on the host health. So CRO will produce the CRO protein, C1 will produce the C1 gene, both are made simultaneously. And depending on the host's health, this can be either by heat shock, cold shock, if there's starvation going on, if a mutagen is present, if there's overcrowding of the cells, will determine which is kept, because different enzymes will be present at different times, different enzymes having different proteolytic activity, meaning lysis of proteins or breaking down of proteins. And so what are the conditions? We'll choose one or the other. If one is chosen, in this case C1 will be chosen and it will block the other one from being made, as you can see here. So the mRNA is being made into the protein blocks CRO from being made. And C1 simultaneously is the gene/protein that prevents the lytic genes from expressing. This is how lysogeny is maintained. In the other example, the CRO wins the race. And so the protein that's made from CRO will block C1 and keep the lytic genes open for expression. Now, lambda's lytic cycle can actually be induced, as we had described previously with our temperate phages, by mitomycin and UV. So if we're here in lysogeny where C1 is being made and it's blocking both CRO and the lytic cycle, under mitomycin and UV, something will happen to the protein. And this protein will no longer be there, and we move on to this one where CRO is now freed up. This is de-repressed. CRO is now being freed up and is able to repress back C1 and keep the lytic genes open to now induce the lytic cycle. This is on a molecular level how that switch looks like. All right, so moving on a little bit, the SOS response is a system that activates in response to DNA damage. The SOS response can be understood based on how it sounds like. So there's many things in science that are complicated to understand based on name, but SOS is pretty self-explanatory, where you're understanding that there's an emergency going on. This mechanism is found across most organisms in some fashion, but it is more conserved from strain to strain in prokaryotes. And it is a global response that addresses DNA damage, which can occur through, as we had just previously described, by UV, by mitomycin, by various chemical mutagens, or even through environmental or physical stressors, physical damage, or cold and heat shock, which are able to damage DNA as well. Now, in this figure that I have quickly came up with, this is just a brief example of how this looks like. So if you have your double-stranded DNA and something happens, the DNA is damaged, you're left, in this case, with single-stranded DNA. And then the protein, RecA, will bind to the single-stranded DNA, stabilize it, and this structure, this RecA binding to the single-stranded DNA, is a signal to the SOS response that something is happening, that repairing is necessary. And so the SOS response kicks in all the genes necessary to repair, and the DNA is repaired back into double-stranded. All right, so RecA de-represses the SOS response by cleaving LexA. So LexA is a protein that, when formed in dimers, will repress the 50+ SOS genes that are present in prokaryotes and prevents them from being expressed. And one of these genes actually being RecA, but a small amount of RecA is produced in the background, just a small basal amount. And so once single-stranded DNA is present, which signifies DNA damage, RecA binds to it and becomes activated. And we have here how we denote activated RecA is a RecA with a little apostrophe. This activated RecA is able to then break apart, let's say, it's able to cleave that dimer, freeing up the SOS genes, and RNA polymerase is then able to make up the proteins, the mRNA, which then turns to proteins that then repair whatever single-stranded or double-stranded breaks of the DNA. RecA has several other roles. In this case, it stabilizes single-stranded DNA, but it is the anti-repressor of the LexA dimer, and it actually is a recombinase, where the rec comes from, and it assists in recombination events to repair DNA. All right, so we've been discussing C1, LexA, and RecA quite a bit already. So to have you understand the research that I'll be showing you shortly, I'll show you how this research was conducted and how this protein was analyzed. So most protein analysis conducted through gel electrophoresis. Historically, within the 1960s through around the 1990s, which most of the research I'll be presenting came from, the gel electrophoresis was essentially the most common tool. And actually the several papers describing how to conduct gel electrophoresis were among the most cited historically, up until, I believe, 2012, last time I checked. And even today, it's still a very powerful tool. And I say 1990s a little bit arbitrarily because we still see it very often today. It's a very simple procedure, relatively speaking, and very easy to do and very cheap to do. But it is around this time that the advent of different molecular tools are becoming cheaper or more available. NMRs, crystallographies, and computer modeling, especially within the last 15 to 20 years, have all become more available and now assist in our understanding of this research. So it's not to say that gel electrophoresis is going anywhere, but now we have other tools that are taking the front seat in these sorts of things. Sodium didecyl sulfate polyacrylonitrile gel electrophoresis, which is often shortened to SDS-PAGE, is gel electrophoresis for proteins. Gel electrophoresis separates things based on size. SDS-PAGE separates proteins specifically based on size. And the way you can read a gel here are that the larger objects, in this case for the SDS-PAGE being proteins, will be on the top, and they will flow down through a matrix towards the bottom, where the smallest are found. So it's almost like a gradient based on size. And this is to separate out different proteins of different sizes. Western blot is a modification of SDS-PAGE, which uses antibodies, which are developed often in rabbits, to select for specific proteins or a specific type of protein. And in that way, if you have, for example, SDS-PAGE with many different bands here, but you want this specific band or this specific protein, you will develop an antibody for that protein, and then you're able to get an SDS-PAGE containing just that protein. It can get far more complicated than that to explain, but for the purposes of this seminar, I believe that should be sufficient. All right, to circle back a little bit, I want to remind you about C1. And here we're showing that C1 is cleaved during an induction event. So during an induction of a lambda strain, the C1 repressor, which represses the lytic cycle and CRO, is being cleaved, this band specifically being the repressor. And then outside of an induction event, this is remaining. So hopefully remember I was describing earlier how when C1 is repressing CRO and the lytic genes, something happens to it to free up CRO and the lytic genes and for the lytic cycle to be induced. And here we're showing experimentally that there's some sort of cleavage, that protein is being broken apart. And we know it's being broken apart because we have a formation of a new band here, R apostrophe. In the absence of an induction, this cleavage does not occur. And so keeping in mind that C1 becomes cleaved during an induction event, that RecA has proteolytic activity where it cleaves LexA to free up the SOS response, and that both an induction of phage and the SOS response both occur in the presence of mutagens like UV or mitomycin C. I formed this hypothesis where RecA mediates the induction of bacteriophage lambda's lytic cycle by proteolytic cleavage of the C1 repressor, and in this case, specifically in the presence of ATP and single-stranded DNA. And why I say this in the end is you will see a little bit later on. All right, so firstly, proteolysis, or the breaking apart of C1, occurs only in RecA positive strains. RecA positive here indicates that there's the gene RecA present, and RecA negative indicates that the host lacks the RecA gene. And lysogen indicates that, or what lysogen means is that that strain contains a phage within it. This induction, which was using mitomycin C to induce it, was conducted with both RecA positive and RecA minus lysogens. And you can see once again that that R band, our C1 repressor, becomes cleaved in RecA positive strains, but nothing occurs in RecA negative strains. So that conclusion that we can make from that is that RecA for sure is associated with proteolysis of lambda repressor. But we're not able to say yet that RecA itself is responsible for it. And so moving on, we can see that C1 is only cleaved in the presence of RecA. So from a strain that is SOS constitutive, meaning that it's always producing SOS response genes or the proteins from those genes, a crude extract was done of all the protein, and it was fractionated, so it was separated out. All the proteins were made into fractions, and each fraction was incubated with irradiated C1. And now you can see that here we have the different fraction numbers, and this is a Western blot of the fractions for RecA. So remember, this is for selecting to identify with antibodies that there's RecA present in these fractions. These fractions were then run with C1, and this is the C1 band. And you can see that proteolysis occurs only when there's RecA present. And when there isn't RecA present, as we see here in 9, nothing occurs. When there's very little RecA, very little occurs. But when we have higher concentrations of RecA in these fractions, we see that the repressor, the C1 repressor, is now being cleaved into 2 segments here. And so we can say here then that RecA, the protein itself, now no longer just a gene, but the protein RecA is now associated with the proteolysis of C1. Why can't we say that it is RecA specifically? Well, because these are still fractions, and so you have contaminants of different other proteins or even DNA in such cases. And this is not just isolated RecA. But then that's how we move on to this next slide, where C1 is cleaved by RecA specifically, in this case, in the presence of ATP. So in vitro incubation, meaning outside of a cell, of purified RecA and C1, with and without ATP, suggests RecA is responsible for the cleavage of repressor. So here we have, and I should start with C. In C, we have just the repressor, and you can see nothing occurs. In lane B, we have both RecA protein isolated, right? Purified and isolated, along with that repressor protein, nothing occurs. But in the presence of ATP, the reaction can be conducted. Now, interestingly enough, in the paper, they were saying that there's a low efficiency, and they were surprised by this. Why do we have so much repressor here and very little proteolytic activity, even though we have everything we need? And they were suggesting that this may be due to the concentrations of the repressor to RecA, that you might have a lot more of one to the other, and that this can affect the protein kinematics. But interestingly enough, further studies suggest otherwise to this. And so moving on to this slide, we have a hypersensitive C1 that is cleaved by background traces of RecA. So here we have C1 ind strain. It's just a strain name that has a hypersensitive C1. It's very sensitive to low amounts of RecA. And we have a C1 plus strain, just named that way, which for this purposes is just a regular lambda strain, or a strain that has lambda-phage in it. And so this hypersensitive C1 strain and this regular C1 strain were compared at different UV intensities. And here you can see for C1 plus, this specific section of the experiment was run at 40 joules per meter squared. And over about half an hour, you can see that there's induction, or sorry, the cleavage of the repressor occurring over that time. Within the C1 in strain, which is the hypersensitive one, within 5 to 10 minutes, almost all of it is cleaved. So that just shows you how it is hypersensitive. But interestingly enough, when you lower the amount of UV to just 2 joules per meter squared, you can see that there's proteolysis still occurring, that C1 is still being cleaved. And just for reference, 2 joules is essentially nothing to a cell because the S-verse response only activates above 16 joules per meter squared. So very little damage is actually occurring here. And again, hopefully remember that RecA is repressed, right? It is part of the S-verse response genes and it is repressed by LexA. So there's no induction of RecA occurring here. There's just a basal amount of RecA, that small amount that's in the background. And that small amount is enough for this proteolysis to occur. And so expanding on what we had just described, where we were describing how basal amounts of RecA is enough to cause proteolysis of the repressor. Interestingly enough, overexpression of RecA does not induce lambda's lytic cycle, which it might be confusing at first because you might think that, well, if RecA is responsible for this, well, how come a lot of RecA is not causing this proteolysis? So 3 strains were compared. A wild type strain containing just the regular lambda-phage, a PBU1 containing strain. So this contains both lambda and the plasmid, which is a control in this case, has no effect on the experiment, or should not have an effect, I should say. And then a strain containing PBU14, which is the same PBU1 plasmid, but with a gene for overexpression of RecA. And so all 3 are compared at these 4 conditions, being at 30 degrees, 40 degrees, being at 40 degrees, and then moved over to 30 degrees, and then having 40 degrees and UV in addition. And here, they're looking for plaque-forming units. So hopefully remember, as I was saying, how they found the lambda phage is where they have it on a plate, where they were studying E. coli on a plate, and they saw little units, little plaques forming, little empty spaces of lysis. And that is what we call plaque. And that's where the phage is induced into its lytic cycle and beginning to kill the cells. And so here they're counting with plaques. In the paper, it was described that these 3 had very little differences. And there's a mathematical equation for statistical significance or insignificance. And these were all shown to be relatively the same. But interestingly enough, in the worst conditions, quote unquote, for the cells, where you have a hot temperature, optimal temperature being around 37, 40 being a little bit uncomfortable for the cells, but you're also blasting them with UV, so you're inducing mutations likely. The wild type strain and the plasmid both had pretty high rates of lytic induction, but the strain that contains the overexpressing RecA plasmid was actually far lower. And now the paper itself does not make a notable or a significant conclusion out of this, and more so is just a report that this is a phenomena that occurs, but this can be related back into how basal or low amounts of RecA are important, and that it is not the induction of RecA that is important. There's something else going on. And what this something else, we're just going to describe right now, is that RecA cleaves C1 in the presence of single-stranded DNA and ATP. So RecA cleavage of C1 depends on ATP and single-stranded DNA. Here we have, in parallel, the same experiment occurring. One has ATP and one has ATP gamma S. This is just a derivative ATP that was coincidentally found or coincidentally tried, and they found that it was far more efficient, but it's just an analog of ATP. And so here we have RecA protein and the anti repressor, sorry, the C1 repressor. With just ATP, nothing occurs. With just single-stranded DNA, nothing occurs. But when both are combined, we have that proteolysis occurring. And this proteolysis is far stronger in the presence of ATP gamma S, where we have both single-stranded DNA and ATP gamma S. Proteolysis is far stronger. So going back to how we were describing the activation of RecA and the cleavage of LexA, it seems here that single-stranded DNA activates RecA, allowing it to cleave C1. And kind of to throw a little curveball, early on I was describing how we were going off of RecA's ability to cleave LexA as a jumping pad to understand that maybe RecA can cleave C1. But interestingly enough, and this shows you how lambda is important to biological breakthroughs, this publication was published in 1980, and using this understanding, they actually found that RecA cleaves LexA in a similar manner. In March 1980, the publication occurs. So that's just a little bit of a reminder how the discoveries made on lambda are then applied to bacteria and why lambda was important for that reason. But in conclusion, essentially for the slide, ATP and single-strand DNA are necessary for the reaction to occur, and that single-strand DNA somehow activates this. And to conclude my presentation, the main points that are covered are that proteolysis of C1 only occurs during an induction of lambda phage with a mutagen, de-repressing the lytic cycle. So the C1 protein was being broken down during an induction when we were using mitomycin or ultraviolet light. This proteolysis only occurred in RecA positive backgrounds, which contained the RecA gene, and the presence of the RecA protein was necessary for this proteolysis to occur. The induction of the gene RecA and the S response genes overall was not necessary for the proteolysis of C1, that the basal small amount in the background of RecA was enough, it was sufficient for the breakdown of C1. And on the flip note of that, overexpression of RecA actually did not induce lambda's lytic cycle. The basal amount of RecA is sufficient, and that the overexpression actually hindered, if you remember the results, it hindered the ability of the phage to become lytic. All right, and the final key to all this was that single-stranded DNA, as well as ATP, was necessary to activate RecA to cleave C1. If you remember early on, I mentioned that they were putting RecA and C1 together, and they were seeing that little proteolysis was occurring. And now we know that it was the absence of ssDNA, and that the RecA wasn't activated. And altogether, this suggests the molecular mechanism of how lambda tunes in to what is happening in the host. Because if the host has its DNA being damaged, which is what kicks off the S response, if there's a threat to the host DNA, this also means that there's a threat to lambda's DNA. And so this is suggesting the mechanism of how lambda tuned in evolutionarily to understand what is happening in the host, whether there's being some harm, whether there's some DNA damage going on, and hijack that system to then be able to know when there's damage occurring and when it's time to leave the cell and move on to somewhere else. And these are the references I cited, and this concludes my presentation overall. And thank you for watching, and enjoy the rest of your evening or Thanksgiving if you watch this on Thanksgiving Day. Hopefully you don't, and enjoy your dinner